The first speaker in the session 2 is Dr. Joseph Powell. He in Garvan Institute of Medical Research. He is currently the Director of Cellular Science at the Garvan Institute and Deputy Director of the UNSW Cellular Genomics Futures Institute. He has played a pivotal role in developing the cellular genomics capabilities and the community in Australia, establishing high-throughput core platforms. He leads a research lab focused at the intersection of cellular genomics and popular genetics. Today's topic of his talk is population genetics meets cellular genomics. Unfortunately, he could not join this symposium lively, so he sent us a pre-recorded video. So we'll watch this video, please.
Hello. Thank you very much for the invitation to present today. I'm very sorry that I have to pre-record this talk. Unfortunately, due to scheduling clashes, I've been unable to present live, so my apologies. My name's Joseph Powell. I'm the Director of Cellular Science at the Garvan Institute of Medical Research, which is the independent medical research institute physically located in Sydney, where I'm joining you from. And it's a pleasure for me today to talk about our work really at the intersection between human genetics, population genetics, and cellular genomics. And I'm going to share with you today two stories of our research that really run along parallel lines to address the same problem, but one using circulating immune cells and the other using induced pluripotent stem cells. And the challenge that we're trying to resolve and to understand is that we already know that there's an enormous number of positions in the genome where there is a segregating genetic variant whose alleles are associated with a complex disease or a phenotype. These have been extensively studied using approaches such as genome-wide association studies. And for most diseases, there's often hundreds, possibly even thousands of these positions across the genome. Now, the vast majority of these loci are located in regions of the genome that do not encode for proteins, typically in regulatory regions. And it's hypothesized and demonstrated with a fair degree of evidence now that the vast majority act through some form of changes of genome regulation. Now, one of the ways that's been extensively used to study these effects and to understand both the way in which human genetic variation impacts just biological differences between people, but also specifically diseases, is through studying what are referred to as expression quantitative trait loci. And this little cartoon at the bottom here is just depicting what I mean by that. And it's really, again, the relationship between the alleles carried at a variant. So in this copy, in this example, copies of the little a allele, and a change in the expression of a gene that's measured, using RNA sequencing, for example. And EQTL studies have been, very extensively used. Have been used to link a large number of disease variants through to function, as well as understanding an enormous amount of biology. And one of the major interesting topics that arose in the last number of years is work showing that whilst the DNA is the same in pretty much every cell and tissue in your body, the way in which that genetic variant can impact the expression can differ in different tissues. And I'm just showing again a sort of cartoon example summarizing some of the work that came out of the GTEx consortium, so a very large international consortium of research, where they've measured the RNA expression in approximately 45 different human tissues and used EQTL mapping to test the relationship between these genetic variants and change in expression, and then asked the very simple question of whether or not these genetic effects have the same allelic effect in one tissue compared to another. And they found plenty of examples like this top one here, where we essentially saw the same effect. So each copy of the little a allele increases the expression of a gene. They found heaps of examples where there's a presence in one tissue and an absence in another one. So genes still expressed, just no variation dependent on the genotype. And then they found a few examples that are really fascinating where there was an opposite direction of effect. So each copy of the little a allele increased the expression in one tissue, decreased it in the other tissue. And this is really where we sort of arrived on the scene with the advent of single-cell sequencing, because this observation, whilst fascinating, also begs the question, well, we know that these tissues are comprised of a whole variety of different cell subtypes. We know that they have very specific functions. They have their own, in many cases, known transcriptional signatures. So if we were expecting to see these differences between different tissues, what would we expect to see at the level of individual cells? And this was the question that we posed ourselves. And really, to solve this, needed to solve a series of both technological, biotechnological challenges, computational methods, before we could really get to the biology. And we've been working on this question from a few different angles. As I mentioned, we'll talk about a study in blood, this one here, and another one using stem cells. And the work in blood started with the creation of a cohort, which we refer to as 1K1K, where we wanted to be able to generate single-cell RNA sequencing data on, the aim was 1,000 cells from 1,000 individuals, hence the name 1K1K. We ended up with a little bit more than that, which was great. So we generated about 1,200 cells per individual, a little over 1,000 individuals. And these were sequenced using the 10x Chromium controller. We also genotyped this cohort, and the genotypes were imputed to haplotype reference panel. And this gave us an experimental design to be able to start looking at EQTL mapping in individual cell resolution. So this work was published a few months ago. We've made all the summary statistics available on this browser, 1k1k.org. All the code is available on Zenodo, and all the data is publicly available on a combination of Geo and the Human Cell Atlas. I'll talk through some of the results, but I wanted to flag a couple of the challenges that I just mentioned that we needed to solve in the process of generating this resource, generating this cohort, and looking at single-cell EQTL mapping. And these were around being able to classify cells accurately. And so we developed an independent cell classification machine learning method, which we call SCPRED, which can be applied to pretty much any single-cell data, which was published a couple of years ago. And the second challenge was being able to deconvolute cells back to original donor, separate them from the types of doublets and multiplets you get in 10x data, and improve the overall recovery of cells. And we have work on a method which we call Demuxify, which is an amalgamation of a series of approaches and software packages that's currently on bioRxiv and will be out in press very soon. So with this experimental design, this gives us the platform to be able to say, right, well, here's a gene that's expressed in one cell type or multiple cell types, and let's test the relationship between SNPs that are within the cis regulatory region and the relationship between the alleles of those SNPs and the changes in the expression. And this is just sort of depicted in this little cartoon at the bottom. Genes expressed in cell type 1, cell type 2. We can test the alleles at a SNP and look at the relationship between those alleles and the changes in expression, and see examples like I described with the tissues, but resolved now to the cellular resolution. So we performed this test. We identified a study-wide significance threshold of about 25,000 of these independent variants, which was an enormous haul. And then we started with this really, straightforward question that I mentioned of looking for the overlap between different cell types. And the upset plot here on the right is basically giving the explanation that the majority of the observed genetic effects are highly cell type specific. There's a few caveats around that, and I'll show a little bit of data to convince you. But at a take-home message, we anticipate somewhere between 65 and 70% of all the genetic variants act essentially in a cell type or cell state specific manner. We're very fortunate to be able to replicate these results using 2 independent cohorts of different ethnic backgrounds through collaboration with Jimmy Yee's group at the University of California, San Francisco. So these plots on the left here, we're just showing the percentage of the reproducing EQTLs that shared allelic direction. And again, just a breakdown across different cell subtypes here on the right. So this was our first approach of being able to resolve the way in which genetic variation acted in different cells, in different cell types. And we made the very deliberate decision early on to stick to cell types that were kind of essentially classified using traditional classification approaches, or known to be understood by the immunological field. But one of the really exciting components of this sort of single cell data, and is personally where I feel a large amount of the field will move in the very near future, is the ability to resolve what we refer to as dynamic effects. So how does a genetic effect vary across some form of cell state or cell landscape as a cell transits in response to a stimuli or a phenotypic response, or a differentiation journey, or so on and so forth? So we chose B cells as our example to really dig into this, and we put B cells agnostically within a manifold space. We could show that cells transited in this space essentially from a naive B cell through to a memory B cell. And then again, we could address the same question, but using dynamic linear and nonlinear regression models to test for changes in the genetic effects as cells transit across that landscape. So this is a different type of biological question to the cell type specific effects. And we're able to identify a set of little over 300 of these genetic effects where we see significant differences in how a SNP asserts its control as a cell transits across this landscape. So these are particularly interesting examples because they essentially give, I guess, a granular example of the way in which genetic variation, whilst fixed in all the cells, asserts its control in a variable manner dependent on the type of state that cell is belonging to. And you can, I think, conceptually take this a step further by thinking if we can reconstruct a cell state landscape that is the direct recapitulation of a biological process, it would allow us to really understand the way in which genetic differences between individuals drive essentially changes in cell behavior and cell function. So just to sort of drill into a few examples to convince you, I think, of the power of this sort of data before I show you how we've been using it to make impacts into understanding clinical translational needs. Here is a series of, I guess, key regulatory cytokine chemokine signaling genes. And I'm just pulling out a few examples, CCR6 and BAC2, which are both genes that are been heavily demonstrated to be involved in both immune cell function and the pathogenesis and of autoimmune diseases. And you can see examples here of how, whilst the genes are expressed relatively ubiquitously in most cell types, the allelic effects are very different in different cells. But very importantly, we can show examples of the way in which different SNPs are associated with the changes in the expression of these genes. So to take BAC2 as the example here, we see a genetic effect of this SNP, the RS729, so on and so forth, which increases the expression of the gene in CD8NC and CD4NC cells, and then other SNPs that control the expression in an opposite directional effect in memory B cells. Now, the fascinating component of this is that these SNPs they're not in linkage disequilibrium with one another. And so this gives very strong evidence there are essentially independent regulatory mechanisms that control the expression of that gene in different cell types. So this is a type of really valuable insights you can get into the way in which natural human variation between individuals really does control cell phenotypic variation and how that varies in cell type to cell type. So circling back to our original problem that we wanted to solve was trying to understand the way in which these genetic effects essentially control or contribute to human disease. And as I mentioned, there's these amazing catalogs of positions in the genome where we've got associations with complex diseases that have come out of things like genome-wide association study. But we want to address and solve a series of essentially interrelated hypotheses. And the first is that there's two variants in the same region, but they're essentially in linkage disequilibrium with one another. So SNPs and LD, one controls transcription, the other a phenotype. It's not unrelated. The second effect, which could be referred to as pleiotropy, is that there's the same causal variant. It has an effect on a phenotype like disease risk, but it also has an effect on transcription, and it's just coincidental. There's no functional relationship between them. And the final hypothesis is that there's a causal variant and it impacts the phenotype, the disease risk, but it impacts it through the changes of gene expression or transcription. In other words, there's a direct causal route by which that variant has an impact. Unfortunately, there's a really nice framework to be able to test for these differences using Mendelian randomization. And so we applied a Mendelian randomization approach to 7 autoimmune diseases that had been very well understood and characterized by genome-wide association studies. And for each of the variants that have been identified where there's a shared loci within one of the single-study QTLs, we can then test for the differences between these 3 hypotheses. And I'm just showing on the right here an example from multiple sclerosis, GWAS, and these columns on the left are the different genes or loci in the genome where there's a known risk-associated variant for MS. And the examples plotted where we've got a colored cell is examples of showing that particular variant acts through the expression of those genes only in these subtypes of cells. So you can see that within the HLA region, unsurprisingly, there's very ubiquitous effects across a large number of cells. But outside the HLA region, these allelic effects, the control MS, act through the changes in gene expression in a very cell-type specific manner. So this starts, I hope, to convince you that we have this ability to map and resolve the functional way in which disease risk acts in different cells. And ultimately then, of course, we're particularly interested in using this to start identifying mechanistic targets that are cell-type specific for new therapeutics. The other really interesting component of the work that we've done is that many of the genes and EQTLs that we've identified are essentially the targets of known immune modulatory drugs. And so the question that we posed to ourselves again was, if there's a variation in the essentials expression of a particular target at baseline for a patient, would that predict whether or not a patient will respond positively or not positively to an on-market drug or a drug that's undergoing clinical trial? Now, unfortunately, for confidentiality reasons, I can't give you the details of these 2 drugs, but these are 2 clinical trials for immunotherapy drugs that both target, as you might anticipate, classic pathways in specific cell subtypes. And we had been conducting these clinical trials for a couple of years and could retrospectively go back and subdivide the patient cohort into individuals based on different alternative genotype classes, where the genotypes are shown to be associated with a relative abundance of the targets of this drug. And you're seeing here just the survival plots on the right, showing the variation in the survival over a 24-month period for the 2 groups of individuals, the individuals with a homozygous genotype that was indicating high target expression, and the individuals with a homozygous genotype group, so it was indicating low target expression. So this is a very early result for us, extremely exciting. And as you can imagine, we're doing an enormous amount of work through to understand mechanistically what's happening, but also to expand this beyond these particular 2 IO drugs. So that's our story to date with using circulating immune cells. But as I mentioned at the beginning, the other parallel arm of this research is to use induced pluripotent stem cells to address the same question, but to do so in cells and tissue types that are basically difficult to access. Blood's very easy to collect from patients, but collecting neurological tissue from hundreds of thousands of individuals is not. And one of the really fascinating components of this is that, again, with the exception of a couple of tissue systems, your body's constantly replacing cells from a pluripotent to mature state. So at any point, cells are transiting across some form of developmental lineage. And as cells transit from a pluripotent to a mature state, they're undergoing transcriptional changes. And you can see where this is leading. We're, of course, then interested in the way in which genetic variation essentially dynamically varies as the cell moves across a developmental lineage. And sorry, this is the slide that was to explain that. And so the way in which we're thinking about this is that we're able to generate a resource of induced pluripotent stem cells, differentiate those cells down multiple lineages, and use genetics again to, and do perform single-cell sequencing longitudinally across those cohorts, and use genetics to be able to unpick the way in which genetic variation controls changes in genome regulation and how that dynamically varies across different lineages. Now, the challenge in doing so is that to practically handle, proliferate, and then, of course, differentiate cells from hundreds of lines, which is the sort of scale of the experimental designs you need, using stem cells is both laborious and expensive, and has a few other challenges associated with batch effects and so on and so forth. So the way in which we've been thinking about this to be able to really massively scale stem cell studies is to use what we refer to as iPSC villages. And a village is basically a co-culture system where we culture lines from multiple unrelated individuals in a single dish. We can differentiate those down lineages, and we can maintain those cultures, and we can treat them with different drugs. And for using single cell sequencing and the assignment of a cell back to a donor, we can extremely easily deconvolute cells back to the donor from a village system. So we have a paper that's currently on bioRxiv, should hopefully be out in press very soon, which describes this village system and addresses the question of whether or not essentially you have the kind of the stem cell equivalent of an allogenic reaction. In addition, without going to the techie details of this, we don't. And there's a couple of additional benefits in that co-culturing essentially reduces batch effects, or otherwise known as line effects, as well as being able to massively scale these studies. So this is our approach where we're building a resource of hundreds of lines from unrelated individuals and both diseased and healthy individuals, and then using directed differentiation and single cell sequencing to then model the way in which genetic variation contributes to cell phenotypes across developmental lineages. Our first paper, just looking at the same question using stem cells that we were looking at in blood, was published last year, where we took fibroblasts from about 80 individuals, did single cell sequencing, and could show the same effects as we saw in blood, where we essentially see quite dramatic differences in the genetic effects and gene expression in different cell subtypes, and then reprogram these cells into induced pluripotent stem cells, and could show, as perhaps unsurprisingly, that the genetic effects were wiped between fibroblasts and iPSCs. But again, you get a recapitulation of differences in the genetic effects acting on gene expression in different cell subtypes of stem cells, and you could dynamically evaluate how they change as cells move from a kind of a quiescent through to an early exit from pluripotency state. And our ultimate view of this body of work is that we are able to identify how genetic effects control, cellular phenotypes such as gene expression, predict how they are likely to impact on actual functional phenotypes, validate that, and then ultimately use these as high throughput compound screening approaches where we've got a diverse genetic background to be able to evaluate pharmacogenetic properties. And the first study that we performed in collaboration with Nathan Palpent's group to demonstrate our approach of doing this was this paper here, published a couple of years ago, where we did single cell sequencing of lines differentiated down a cardiomyocyte lineage, and we sequenced them at multiple time points, and we could predict and identify subpopulations of cells and what transcriptional signatures were unique between those populations and what genetic variants influenced that. And the first study shown on the right here shows these different populations of cells, and we could predict as cells matured to mature cardiomyocyte stage, there was a genetic effect on a transcription factor network that we predicted initially to be impacting the contractile forces of the cells. So we went back into the cell population, we sorted the mature cardiomyocytes based on the cell surface signals identified from the RNA sequencing data, and then we used a 3D organoid model where we could measure the contractile forces of the cell populations and could show that the cell population A had a different contractile force to cell population B as predicted from the original data. So this is, I guess, my way of hopefully convincing you that we're able to use the intersection of population genetics, stem cell technology, and cellular genomics to be able to resolve the way in which genetic variation really functionally acts as cells move from a pluripotent to mature state. And we've been scaling these studies. I suppose we're still in the foothills of these bodies of work at the moment, but we've been scaling these studies, taking these cohorts of iPS cells and then differentiating them down different lineages. And the first sort of major directed village-based differentiation we've had is down the retinal epithelial and retinal neural lineages to study how genetic effects that underlie glaucoma and age-related macular degeneration, which I'm not showing here, functionally resolve in different cell types. So again, I don't have time to go through all the details of this, but we use very similar experimental designs to what I've been talking about, using a large population of stem cells, differentiated down the lineages, use single-cell sequencing, could map how the genetic effects varied in different cell types, and then functionally link that, in this case, through to protein expression in different cell types, and show how the genetic risk variants for glaucoma and AMD likely functionally acting in different cell subtypes. So I'm going to finish up there, and hopefully that's been a coherent explanation of our body of work of intersecting these various fields. I'd like to thank you all for your attention. Again, I'm sorry for not being able to present live and answer questions, but also I'd like to really acknowledge a fantastic group that I work with and that you can see here on this long table, and call out the work from Seyhan and Jose, who really led a lot of the 1K program of work, and Drew and Anne, who have led most of the work that I mentioned in the stem cells, and just acknowledge really fantastic collaborators in both Australia and the States. And thank you very much for your attention.
Thank you, Dr. Powell. As I mentioned, he sent us a pre-recorded video, so we do not have any Q&A session. So if anyone has some question, you can send an email to Dr. Powell, or send us an email to us. Then we will transfer the email.